Salmonella typhi is a gram-negative motile, capsulated and rod-shaped bacterium. The colonies do not have a particular arrangement, and each cell measures between 0.6 to 0.8 micrometers wide and between 2 to 4 micrometers in length. In humans, infection with Salmonella typhi results in typhoid fever. The clinical incubation of the disease ranges from 5 to 7 days. A very important risk factor to acquire this disease is the diminution of the intestinal microbiota by the overuse of antibiotics or by undernutrition. The pre-pathogenic period of the disease comprises the transmission from an infected person to a healthy one through the fecal-oral route. The entrance of the bacteria through the gastrointestinal tract may happen by the consumption of contaminated water, undercooked foods, contact with fomites of infected patients, and is more common in areas with overcrowding and poor sanitation. The pathogenic period begins with asymptomatic gut invasion and dissemination of the bacteria. In the first week, the most common symptoms are nausea, vomiting, diffuse abdominal pain, bloating, anorexia, and diarrhea, followed by a short asymptomatic phase that gives way to bacteremia and fever with flu-like symptoms, accompanied by a decreased heart rate, followed by arthralgia and enterocolitis. In the second week, findings include abdominal distension and, when ileal perforation happens, tenderness, rigidity, and guarding of the abdomen. The patient looks pale, mildly distressed, and dehydrated with sunken eyes, dry skin, and lethargy. In the third week, without treatment, the patient becomes critically anorexic. Chances of bowel perforation increase, which worsens abdominal distension and peritonitis. The patient becomes tachyphenic, with crackles over the lung base and oscillation. Signs of metastatic complications appear, such as dry cough due to pneumonia and neck rigidity due to meningitis, or even chest pain due to myocarditis and pericarditis. Stool shedding and transmission may occur during the acute symptomatic phase of the disease, and up to 4% of the patients who recover from it could evolve to be chronic asymptomatic carriers of Salmonella typhi, which results from long-term gallbladder carriage. For the detection of Salmonella typhi, its distinctive virulence factors are targeted, one of those being the O antigen, part of the lipopolysaccharide, which has 3 regions that define serotype specificity. Its strong endotoxin activity triggers inflammation and can lead to severe symptoms. Then there's the H antigen, made up of flagellin proteins, which drives movement and enhances virulence. Finally, the VI antigen forms a protective polysaccharide capsule that aids in the evasion of immune cells. Other targets include HIL-A, a transcriptional regulator critical for virulence, which controls the expression of virulence factors essential for host invasion and survival, and the 16S rRNA gene, which is essential for protein synthesis. It also focuses on FlyC, the flagellin gene, and SPI1 genes such as INF-A, which is part of the type 3 secretion system, and CYP-A, both of which aid in the invasion of host cells. SPI7, which carries TBI-A and TBI-B genes, is also considered, because said genes contribute to host adaptation and survival within macrophages. Nested PCR is a technique developed to increase the sensitivity and specificity of standard PCR reactions. The master mix contains DNA polymerase, DNTPs, buffer, magnesium chloride, and 2 sets of primer pairs. The first set amplifies a region slightly larger than the region of interest, while the second one targets the specific region of interest, ensuring the specific amplification. Multiplex PCR allows the simultaneous detection of multiple targets using different primer sets in a single reaction, increasing efficiency and differentiation. The setup involves using template DNA from different sources, with multiple forward and reverse primer sets tailored to each target. Now, multiplex nested PCR is a variation of PCR in which 2 or more loci are amplified simultaneously in a single reaction, thereby improving the reliability of detection. It combines the ability to detect multiple targets of multiplex PCR with enhanced sensitivity and specificity of nested PCR. The test can detect Salmonella typhi and Salmonella paratyphi, particularly for the MDR H58 and XDR lineages, which deems it as a confirmatory test. As for the sample to be used, it is usually a blood sample obtained from patients suspected of having typhoid fever. DNA from blood samples is extracted from 0.5 to 1 milliliters of blood containing 20 millimolar potassium EDTA. The sample is centrifuged at 10,000 RPMs for 5 minutes. 1 milliliter of lysis buffer is then added to the pellet. The mixture is then gently aspirated multiple times to produce hemolysis, to then centrifuge the tube at 12,000 rpm for 6 minutes. The supernatant is discarded and the pellet is washed again with the lysis buffer followed by distilled water. Finally, the pellet is resuspended in 20 microliters of distilled water. The tubes are then sealed and subjected to lysis in boiling water for 20 minutes and brought back to room temperature before being used as a template for PCR. In the first multiplex PCR step, a DNA template is added to a reaction mixture containing tris HCl, potassium chloride, magnesium chloride, gelatin, specific primers, deoxynucleoside triphosphate, Taq polymerase, and distilled water. This mixture is subjected to a series of temperature cycles, including denaturation at 94 degrees Celsius for 5 minutes, followed by 30 cycles of amplification at 94 degrees Celsius for 30 seconds, 55 degrees Celsius for 30 seconds, and 72 degrees Celsius for 1 minute, with a final extension at 72 degrees Celsius for 5 minutes. In the second nested PCR step, a portion of the previous PCR product is added to a new reaction mixture similar to the first step, with some adjustments to the temperature cycles, including an initial denaturation at 94 degrees Celsius for 5 minutes, followed by 30 cycles of amplification at 94 degrees Celsius for 1 minute, 50 to 60 degrees Celsius for 30 seconds, and 72 degrees Celsius for 1 to 2 minutes, with a final extension at 72 degrees Celsius for 5 minutes. The virulent strain of Salmonella typhi, MTCC734, is often used as a positive control for this test. The bacterial DNA from obtained cultures is extracted with chloroform. 2 ml of overnight cultures are taken and centrifuged at 6,000 rpm for 10 minutes at room temperature. The pellet is resuspended in 0.5 ml of 1x-STE by vortexing for 2 minutes. Then 50 microliters of 10% SES and 150 microliters of 5 molar sodium chloride are added to then make a proper mixing. After that, an equal volume of chloroform, isoamyl alcohol mixture is added, followed by centrifugation at 12,000 rpm for 10 minutes at room temperature. To the aqueous layer, 1/3 volume of isopropanol is added and centrifuged at 12,000 rpm for 10 minutes at room temperature. The resulting DNA pellet is air-dried, resuspended in 20 microliters of sterile water, and incubated in a water bath maintained at 37 degrees Celsius for 3 hours or overnight. For long-term storage, DNA can be resuspended in 20 microliters of TE buffer and stored at -20 degrees Celsius until its use. As for the determination of the sensitivity of the multiplex nested PCR test, 0 dilutions of DNA can be made employing sterile water for PCRs, making it so that the PCR can detect Salmonella typhi at a determined number of colony-forming units per milliliter. The primers employed are specifically designed for the flagellin gene, FLY-C, and VI-negative genes, TBI-A and TBI-B. Based from these, a determined applicon is expected to be formed, which is measured in the amount of base pairs it has. A positive result is confirmed by observing the amplified DNA bands on an agar gel. For example, as we can see in this image, the use of a single primer can yield products of 363 BPs, 307 BPs, and 774 BPs, which therefore confirms the presence of genes FLY-C, TBI-A, and TBI-B, respectively. These can be seen in samples 1 to 4, while in samples 5 to 9 there is seemingly no formation of those. However, on sample 8, very faint stripes can be seen on the gel, so this could be considered as a doubtful result. One of the great advantages of multiplex nested PCR is the high specificity and sensitivity for the detection of Salmonella typhi when compared to other methods. This test can identify lineages of low or high risk within a single test, and the detection of the pathogen is also quicker compared to other methods. While this test can effectively detect Salmonella typhi, it also identifies Salmonella paratyphi, which is an occurrence that could interfere with the diagnosis. Its usage is constrained by cost given the high specificity of oligonucleotides or primers employed. Furthermore, it demands meticulous handling, thereby increasing the risk of contamination. As of today, one of the laboratories that sells something close to this test here in Mexico is Emunet. They sell test strips specifically designed for the detection of Salmonella spp. Each box is available at a price of 1,900 pesos. That's all for today from Team 12. Thank you for watching.